In this video, we learn more about the steps involved in the process of gene cloning. In this image of a petri dish containing nutrient media conducive to bacterial growth, you can see that there are tiny white colonies growing on the media. Each of these colonies contains thousands of bacterial cells, and these cells contain multiple copies of plasmids. Plasmids can serve as vector DNA to carry the gene of interest in the process of gene cloning. The gene of interest is isolated from one of a variety of sources, for example, a human skin cell. The gene of interest and the vector DNA are digested or cut using restriction enzymes and combined together to form a recombinant vector, which is then inserted back into bacterial cells so that multiple copies of that vector can be generated as the bacteria multiply. So, to carry out the process of gene cloning, you need the vector DNA, which acts as a carrier for a DNA segment to be cloned, which from now on we'll call as the gene of interest. It is often a plasmid, as I stated before, or it can also be viral DNA. You need restriction enzymes to be able to cut DNA at specific sequences, both within the plasmid as well as around the gene of interest. As I mentioned in an earlier video, there are over 3,000 different enzymes that have been so far identified in microbes. You also need an enzyme called DNA ligase, which ligates or seals the sugar phosphate backbone of DNA fragments. This enzyme is used to ligate the gene of interest with the vector DNA or the plasmid. The first step in gene cloning is to cut the DNA with restriction enzymes. So the vector DNA and the DNA containing the gene of interest are digested with the same restriction enzyme. Now, this is important. The same restriction enzyme needs to be used because we need to generate sticky ends that are going to be complementary to each other. In our example here, the enzyme is EcoR1 and it recognizes the sequence GAA-TTC within DNA. Now note the different features of the vector DNA: the LAGZ gene, the ampicillin resistance gene, and the origin of replication. We're going to talk about these features in a few minutes. But right now, focus on the digestion with the restriction enzyme of the vector DNA as well as the gene of interest, producing sticky ends that are complementary to each other. The next step is to add ligase to this reaction, in which both the digested vector DNA and the digested gene of interest are combined in the same reaction. The sticky ends produced by the restriction enzyme hydrogen bond with each other, and the ligase helps seal the DNA backbone of the recombinant vector, thus making the circular DNA complete again. The next step in gene cloning is the transformation of bacteria with the recombinant vector. So the recombinant vector is added back to the bacterial cells, which take up the DNA through pores in their membranes. And these pores are created by adding agents like calcium chloride or electroporation. These are treatments done to the bacterial cells to make them competent to receive or take up the recombinant vector. After the vector has been introduced into bacterial cells, they're allowed to multiply, thus generating multiple copies of the gene of interest within the recombinant vector. The origin of replication in the plasmid allows the plasmid to be copied multiple times, generating large quantities of that plasmid and in turn the gene of interest. The next step is selecting transformants, which are bacteria containing the recombinant vector. When we do that, we have to ask the question, how do we differentiate between cells that have successfully taken up the plasmid from those that haven't? This is done by growing the transformation mixture on nutrient growth media containing the antibiotic ampicillin. This is where the ampicillin resistance gene in the plasmid plays an important role. The bacterial cells that have taken up the plasmid, the recombinant vector, will be able to grow on media containing the antibiotic ampicillin. And the cells that have not taken up the plasmid will not be able to grow on this media. Now, it's not enough to tell apart the cells that have taken up the recombinant plasmid from the ones that have not. We also need to know whether our gene of interest has inserted itself into the plasmid vector. So to do that, in addition to adding ampicillin or an antibiotic to the nutrient media, we also need to add an indicator that's going to tell us whether the cells have taken up the gene of interest contained within the plasmid vector. So to do that, we grow the cells on a nutrient media that has the antibiotic, which in this case is ampicillin, in addition to a compound called XCal. Now, the interesting thing about XCal is that it is a substrate for an enzyme called beta-galactosidase, encoded by the LAGZ gene. And we talked about the LAGZ gene earlier in that it was a selectable marker to determine whether the bacterial cells have taken up our plasmid or not. So this is where it comes into play. This is where the gene comes into play. Now we have a LAGZ gene that encodes beta-galactosidase, beta-galactosidase converts XCal to a product that's blue in color. So this product is blue. Now, a functional LAGZ gene will encode beta-galactosidase, will lead to the production of a functional enzyme that will convert our substrate into a blue product, allowing the colony to take on a blue appearance. So all of these blue colonies have bacterial cells that have a functional copy of the LAGZ gene. Now remember that we had designed our plasmid to actually break up the LAGZ gene to take in the gene of interest. So the LAGZ gene basically is disrupted and becomes non-functional. So if the recombinant vector has our gene of interest, then the colony will appear white because the LAGZ gene will be disrupted. So the colonies that we're interested in are the white colonies, because the very fact that they cannot lead to the processing of XCal to a blue product indicates that that LAGZ gene has taken up the gene of interest. So those are the colonies we want. So this is how we tell apart the bacterial colonies that contain cells which have taken up both the plasmid as well as the gene of interest. So if you look at a picture of this nutrient media plate, you'll see the blue colonies that contain the plasmid, but those plasmids do not contain the gene of interest. Whereas the white colonies are the ones that contain the gene of interest. So to summarize the whole process, we started with the gene of interest and the pre-designed plasmid vector, processed the two, combined them, and inserted the resulting recombinant plasmid into bacterial cells. And then we used different ways to determine whether those bacterial cells contain plasmids that carry the gene of interest or not, using several indicators, including the antibiotic resistance gene as well as the LAGC gene, which is the selectable marker. Gene cloning has several applications. These include making multiple copies of a gene of interest, whether it is to make large quantities of the protein that is encoded by that gene, for example, insulin, or DNA fingerprinting, or to study the function of an unknown gene. The process can also be used to transfer a gene from one organism to another. This is the area of transgenics or genetic engineering. And it can also be used to replace a defective gene with a functional copy in patients suffering from certain genetic disorders. Hope this helps. Thank you.